And the first speaker will be Marilina de Sautu from University of Buenos Aires. So Marilina, can you share your screen with us?
Yes, of course.
So you have 15 minutes, okay?
Okay. Can you see now?
Yes.
Okay, thank you. Hello, everyone. My name is Marilina de Sautu, and I am a PhD student from the University of Buenos Aires. And I am going to present this work that I made under the direction of Irene Mangialabori and Mariela Ferreira Gomez. And it's about the influence of the lipid environment in the plasma membrane calcium ATPase activity. So P-type ATPases are essentially primary active transporters of ions, polyamines, and phospholipids. There are 5 subfamilies, and among others, in the type 2, includes the sarcodoplasmic reticulum calcium ATPase, or SIRCA, the sodium potassium ATPase, and the plasma membrane calcium ATPase, or PMCA. These proteins are highly similar, and the transport mechanisms of these PATPases involves the hydrolysis of ATP and the autophosphorylation of the proteins that leads to the translocations of ions against its gradient concentration. PMCA is located in the plasma membrane of all eukaryotic cells. The isoform 1 and 4 are ubiquitous, expressed in all the human cells, and 2 and 3 are tissue-specific. PMCA function is to transport calcium out of the cell against its electrochemical gradient. The only structure known up to date is in absence of calcium and was solved by cryo-EM. This structure is similar to other PATPases. It has 10 transmembrane segments and 3 cytoplasmic domains. The phosphorylation domain, the nucleotidic binding domain, and the actuator domain. Unfortunately, the regulatory parts of the protein that are different from other PATPases were not solved in the structure, and they are the C-terminal autoinhibitory domain and the acidic phospholipid binding domain. There is a general function mechanism for all the PATPases, and it is based in the equilibrium between the E1s and the E2 conformations. For PMCA, in absence of calcium and ATP, the protein is in an equilibrium between the E1 and the E2 conformation shift to the E2. In presence of calcium, it can bind to the calcium binding site that in the E1 conformation is oriented to the cytoplasm and has high affinity. So the protein can bind calcium and then ATP and autophosphorylate, leading to the E1P conformation. In this one, the phosphate is bound to the aspartic acid residue that is conserved in all the PATPases. This conformation is unstable, and through a conformational change leads to the E2P conformation, that in this one, the calcium binding site is oriented to the extracellular medium and has low affinity, so the calcium is released, and then the phosphate and the protein can start a new reaction cycle. Different inhibitors were used to stabilize different conformational states of other PATPases. For example, SIRCA structure was obtained in different states, for example, in the E2 conformation in presence of topsy-argine, in E1 conformation in presence of calcium, in the E1P using aluminum fluoride and ADP, and also metal fluoride complexes like beryllium fluoride, aluminum fluoride, and magnesium fluoride were used to stabilize the dephosphorylation process in SIRCA and obtain the structure. We found that metal fluoride complexes also inhibit PMCA, but aluminum alone, in absence of fluoride, also inhibit PMCA and was not characterized before. So we can measure the activity of this purified protein by measuring the phosphate release by time by a colorimetric reaction. So in presence of saturating concentration of magnesium, calcium, and ATP, we measure the phosphate release by time in absence and in presence of increasing concentration of aluminum. And from the slope of these linear curves, we can calculate the calcium ATPase activity that decreases hyperbolically as a function of the concentration of aluminum. By fitting a decreasing hyperbola function to the experimental data, we calculate the apparent inhibition constant that corresponds with the concentration of aluminum that reduced 50% of the initial activity and was 8 micromolar. Then we can measure the phosphorylated enzyme of the PMCA by using radioactive ATP. In this assay, we are measuring the amount of the conformations E1P and the E2P. And we found that the phosphorylated enzyme increases as a function of aluminum. And the apparent constant of this process was very similar to the one that we found in the calcium ATPase activity, indicating that this is the step involved in the inhibition. So we found that aluminum is stabilizing the PMCA in one of these conformations, E1P or E2P. So the next aim was to study the influence of the surrounding bilayer in this process and in the protein activity. Most of calcium transporter and membrane proteins can be modulated by lipids due to a direct interaction with non-annular lipids or due to an interaction with the first shell of annular lipids. Also by the regulation of the membrane biophysical properties, for example, packing, thickness, and curvature. It was described before for PMCA by Pignataro and collaborators that an increase in the thickness of the membrane affects protein activity because some charges, amino acids, can come into contact with an hydrophobic environment, and this inhibits the protein. As aluminum can interact with the lipids, we used the example of this inhibitor to study if the changes in the lipid environment or the changes in the protein due to the lipid due to the environment can affect the inhibition of aluminum. So for these studies, we reconstitute the protein in different molar fractions of the DMPC, dimeristoic glycerophosphocholide, that is a synthetic phospholipid. So we combine different concentration of detergent. From all detergent, we decrease the concentration of detergent and increase the concentration of the lipid to form the different DMPC molar fractions. And we measure the calcium ATPase activity in presence of saturating concentration of substrates as a function of the different molar fractions. First in absence of aluminum, here in gray, and then in presence of increasing concentration of aluminum. We can see here that in absence of aluminum, the protein is inactive in the presence of all detergent. As we increase the lipid concentration, the lipids can intercalate in the hydrophobic part of the protein, and the protein of the activity increases. And then after a molar fraction of 0.4, the activity started to decrease. We thought that this effect could be due to a transition in the organization of the amphiphils in solution, because the detergent molecule has a cone shape that is more stable, forming spherical micelles in solution, while DMPC is more stable, forming bilayers. Then, in the presence of aluminum, we saw two types of effects. At lower lipid concentration, aluminum can inhibit the pump in all the concentration that we tested. But at higher lipid concentration, we saw that in the presence of aluminum, the decay in the activity that we saw in the control was shifted due to the presence of the inhibitor, and this was seen in all the concentrations that we tested. And in some conditions, in the presence of aluminum, the protein has more activity than the control. So For study these differences between the particles in the different molar fractions, we perform a small angle X-ray scattering experiment in collaboration with the group of the Professor Rosangela Itri and Dr. Gustavo Scarabacchi in the University of São Paulo. So SACS is based in the scattering of X-ray produced by the electrons in the sample. So we obtain the scattering curves of intensity of scattering as a function of the scattering vector in the different molar fractions. We notice that we have two types of scattering curves. At lower molar fractions, we have this type of curves, and at higher molar fractions, 0.4 and 0.6, we have these curves that have different shape. So we analyzed the curves by using the GenFit software, and the best fitting for the experimental data was a cylinder with hemispheres model for the lower molar fractions. And then, as it was expected, when we increased the lipids at the higher molar fractions, the best fitting was the bilayer model. So we confirm here that we are having a transition in the organization that is affecting the activity of the protein. From the parameters obtained by the model analysis, we calculated the electron density of the particles. The electron density corresponds to the electrons per volume, and we plot here the electron density as a function of the distance from the center of the particle towards the solvent in both directions. So in this course, we have an electron density that corresponds to the hydrophobic tails and an electron density that corresponds to the polar heads. And we found that the thickness of the cylinder of all detergent was 64. Then when we add the lipids to form the first molar fractions, we have an increase in the thickness, maybe due to a disorder in the polar heads of the lipids in presence of high concentration of detergent, and this was seen also by molecular dynamic simulations. And then when we increase the molar fraction, the electron density of the polar heads is more homogeneous and the thickness decreases. And in this condition, the protein is reaching the maximum of the activity. We obtained also the electron density profile for the bilayer model, and we found that the thickness does not change too much in the higher molar fractions, but we have an increase in the electron density at higher molar fractions. And this could be due to a more packing arrangement of the molecules in solution. One way to study lipid packing is using the Laurdan probe. So this probe can intercalate in the membranes and can sense the polarity of the membrane environment, that the polarity decrease in more packed membranes. So the probe has this naphthalene group here, that its emission is shifted due to the dipolar relaxation of the solvent, and this can be measured calculating the general polarization. So we plot the general polarization value as a function of the different molar fractions, and we saw that this value increases as we increase the concentration of lipid or the molar fractions, indicating that the intensity of the emission fluorescence of the probe was shifted to the blue, and this means that the probe is sensing less water molecules. That means that in this condition, the membrane is more rigid. It has been described for other ATPases that an increase in the lateral pressure in more packed membranes decreases the protein activity because the conformational change of the dephosphorylation process is affected by this lateral pressure. So this could explain the decrease in the activity that we saw at higher molar fractions. Then, for analyzing the effect of the aluminum on the lipid packing, we performed the same experiments in the presence of aluminum. Here I show you the curve obtained in the presence of aluminum in gray, and we notice that this curve is not significantly different from the one obtained in absence. So this we thought that this is maybe because the laudan is sensing inside the membrane near the glycerol group, but aluminum is most likely to have an effect in the polar heads. Or maybe we are not detecting this because we are doing steady state fluorescence experiments. So we choose to perform the same experiment using a merocenin probe. This is an amphipathic probe that intercalates in the membrane near the polar heads, and the probe intercalates as a monomer in more fluid membranes, and it can form dimers when the membrane is more ordered or rigid. And the emission spectra of this probe has two maximum, and one corresponds to the monomeric form and the another to the dimer form. So the equilibrium between these two forms depends on the packing of the membrane. So the relationship in the emission of this maximum is used to study the membrane packing. So we measure the emission fluorescence of merocenin and in all the molar fractions, and we calculate the relationship of the fluorescence intensity. And we notice that at lower molar fractions, this relationship does not change. And when we increase the molar fraction, we can see a decrease in this relationship. That means that more dimers are able to form and the membrane is more rigid. The same result that we'll obtain with laudan in this part. In presence of aluminum, we notice that at higher molar fraction, the rigidity of the membrane decreases, indicating that aluminum is having an effect probably due to an interaction with the polar heads. And this produces an effect in the protein activity at higher molar fraction. So the conclusions are that we found that the protein activity largely depends on the different molar fractions. Both the thickness and lipid packing have effects on the protein activity, and the aluminum emission was modified through an interaction of aluminum with the surrounding lipid environment. We are working now in a model that relates this effect with the protein structure, and this study showed the importance of the surrounding bilayer in the studies with purified membrane proteins, because the lipid environment can change the protein conformation, but also the inhibitor effects. I want to thank my group in Argentina, my PhD advisor, Irene Mangialabori, my co-advisor, Mariela Ferrera Gomez, and Juan Pablo Rossi. Of course, the University of Buenos Aires for allowing me to perform my PhD, and the collaborators of the University of San Pablo that they teach me a lot of sax, Professor Rosangela Itri and Gustavo Scanamaki. Of course, the funding, and thank you for listening and inviting me here.
Thank you. Marilina, are there questions from the audience? Someone wants to raise their hands or write their questions? Yeah. Please. We have Tony Watts, please. Can you make a question?
Yes, hello, and thank you for your talk. So lipid-protein interactions have been an interest of mine for many years, and in particular with the ATPases. So remind me what kind of structures the C12E10 form at low DMPC concentrations. Are they micelles, or are they bicells, or are they bilayers? And at what proportion of DMPC do you start to form a bilayer?
Yeah, the first ones at lower molar fractions, they are cylinder micelles with hemifers at the final of the micelles, and then at 0.4 molar fraction of DMPC, we start to see the bilayer.
Okay, so at 0.4 molar fraction, you start to get bilayers. So do you think this is the most important part of the activity measurements, that the bilayer is important to the ATPase rather than in a mice cell? Do you think that is the major driving factor for your results?
I don't know if it is the structure itself. I think it has to be the lateral pressure of the lipids and of course the thickness of the membrane, because we perform the same experiments using a mixture of lipids and combining with the same detergent. And if you use a mix of lipids, you cannot see the decay in the activity at higher molar fractions. So it has to be something with the thickness, of course, of the membrane, of the bilayer of DMPC. And because DMPC is all saturated lipids, and we think that the lateral pressure in these bilayers is more than if we have a mixture of lipids, and that is affecting the protein activity.
Okay, and on one of your slides, you had POPC interaction with the ATPase. Was that from some published work, or was that your work? Or did you do everything with DMPC?
No, that's a work with the CIRCA. It's a published work.
Right, sure. Okay, the CIRCA work, of course, has been worked on for a long time, of course, yes. Okay, thank you very much for your talk.
Thank you for your question.
Next, there is also a question from Emerson. Emerson, can you make your question, please?
Hola, Marilina. Thanks for your talk. It was very interesting results. I wonder, what do you think about the implications of your results in the current structures that you have of this protein? Like, or do you expect that with lipids the structures change, or it's more like a dynamic change that you are seeing? Like, I would like to know what is your thinking about it.
Oh, thank you for your question. Very interesting. I think that we are working now in a model. We want to reach which is the best thickness of the protein, the best thickness of the bilayer for the protein activity, and relates that with the structure, because there are several charges residues that they are in the interface between the hydrophobic and the hydrophilic part of the membrane. So we want to relate the thickness, of course, of the bilayer, and think what happened with the activity when these charges, amino acids come into contact with the hydrophobic part, for example, if the activity increases or if the activity decreases. Also, we have the example of the aluminum that clearly is having an effect. What is this effect? It's only, we are trying to answer if it is only on the packing, and why, which is the best packing for the membrane that allows the conformational change. And yeah, we are working in that model now.